Hello, everyone. Can you hear me?
Yes.
Today, I will be sharing with you our latest research on clotting pathologies, and I will spotlight our recent findings in COVID-19 and focus on long-COVID. Here are just some of my collaborators and co-authors of data that is going to be presented here. More on our broader global collaborators later. I just wish to point out Professor Douglas Kell. He's from the University of Liverpool, and he is a systems biologist. And then the clinician, Dr. Jaco Loubser, who is also in the audience. He is a cardiologist from Mediclinic Stellenbosch, and a whole team of researchers, also data scientists from Stellenbosch University. My lab and our collaborators from various labs over the world have been identifying inflammatory molecules in circulation that might be involved in and drive pathological clotting. And we have also focused our research endeavors on studying the effects of increased circulating inflammatory molecules and how they interact with cells of the hematological system. We focus in particular on platelets and red blood cells, as well as on the clotting protein, fibrinogen. We are also interested in identifying novel inflammatory molecules that might play a role in the persistent symptoms in long COVID. Focusing on platelets for a minute, here is a cartoon to show all the various receptors on platelets and their interactions with inflammatory molecules. They also bind to each other and to damaged endothelial cells. I am particularly interested in platelet signaling and the role in abnormal clotting. Platelets in circulation play a critical role in healthy blood clotting. However, they can become overstimulated and can drive pathological blood clotting if there are inflammatory molecules in circulation. This can also happen in the presence of viral infections, where platelets can act as important signaling entities. There is a complex relationship between receptors on platelets and endothelial cells, where these circulating biomarkers may bind to. As we now know, damaged endothelial cells and platelet hyperactivation are central pathologies in acute and long COVID. Our research group have shown these pathologies in both of these diseases in published papers. I just quickly want to focus on some science. If you focus your attention to the two cartoons shown in figure A, on the left is a healthy protein structure, which may have very many alpha coils and few beta sheets. However, in the presence of inflammation, oxidative stress, or circulating inflammatory molecules, these structure changes where the alpha coils untwist into beta sheets, as seen in A in the cartoon on the right. Previously, we added thrombin to plasma to create extensive fibrin clots. This process mimics clot formation in vivo. In plasma clots from healthy individuals, these clots have more alpha coils, with the ultrastructure shown in figure B. These clots look like a net and is typically formed, for example, when you cut yourself. Various inflammatory conditions, including cardiovascular disease and diabetes, have a clot structure, as seen in figure C, where the fibrin net is plate-like and dense due to fibrinogen that have been exposed to circulating inflammatory molecules. Damaged fibrinogen molecules that clot is in this manner, clots in an anomalous manner. Already in May 2020, we, together with our clinical collaborator, Dr. Jaco Loubser, found significant coagulation and platelet pathology in acute COVID ICU patients. It was then that we discovered that fibrinogen formed these spontaneous microclots in circulation. Here you can see scanning electron microscopy micrographs where these fibrin deposits attach to red blood cells. This is simply in a blood smear, nothing added. So early in 2021, we turned our attention to long COVID. We looked at blood samples from patients and soon realized that there are persistent microclots and widespread endothelial and platelet dysfunction in these patients too. These pathologies may be central in causing the widespread symptoms in these patients and may lead to tissue hypoxia. We initiated a South African long COVID registry in 2021. And here are just some of the symptoms noted in these long COVID patients, very much similar to that was reported globally over the past few months. And here's brain fog, concentration issues, forgetfulness, constant fatigue. Those are the symptoms we also report in South Africa. Most prevalent comorbidities are high blood pressure, high cholesterol, rheumatoid arthritis, and type 2 diabetes. We also looked at blood samples from these patients and compared results to samples from healthy individuals. On the left, you can see platelets from healthy patients, and on the right, platelets from patients suffering from long COVID. Large platelet clumps can be seen. We also planned an experiment where we looked at proteomics, where we find proteins trapped inside these microclots, and we compared healthy plasma versus type 2 diabetes, acute COVID and long COVID. We added the diabetes samples as we know that people suffering from diabetes are more prone to severe COVID symptoms. For these proteomics analysis, we prepared plasma and we followed a first trypsin digestion step. To our surprise, we found a visible deposit at the bottom of the tube in acute COVID as well as long COVID, but not in the diabetes and also not in control samples. Plasma proteins, therefore, were fully digested in controls and diabetes, suggesting that trypsin could degrade all, or at least in most of the proteins in the controls and the diabetes. For proteomics, we then filtered the sample and we also digested it further and then we could indeed solubilize the undigested pellets. When we viewed the unfiltered supernatant after the first digestion step using fluorescent microscopy, we noted that patients with type 2 diabetes and plasma from healthy patients digested fully and we couldn't find any signal. The signal we are referring to is the green signal. And you could see that in the micrographs on the top left. In contrast, there were still numerous microclots in plasma from patients with both acute COVID and long COVID. After the second digestion step, we then could digest the microclots and we searched for molecules. We detected various inflammatory molecules that were substantially increased inside the microclots of the COVID and acute COVID and the long COVID versus the equivalent volume of the fluid in the controls. Of particular interest was a substantial increase of alpha-2 antiplasmin, fibrinogen chains, as well as von Willebrand factor. And here's just a diagram for the clotting cascade, and not going into much detail, but look at where alpha-2 antiplasmin has an effect. It prevents clot breakdown. So the lack of breakdown of these persistent clots may have significant clinical value. Here is a simplified diagram of our take on explaining clot formation and then also breakdown. And if you look at this diagram, during acute COVID-19, spike protein induces the microclots that are more resistant to clot breakdown, as we have discovered. But in some individuals, during the recovery process, healthy fibrinolysis will happen as it should, and these individuals will then return to health. However, in some individuals, we now know 10 to 30%, healthy fibrinolysis will not happen. A lack of breakdown of fibrinolysis in long COVID patients may result in a persistent failed fibrinolytic system. We just completed a larger proteomic study where we looked at 100 long COVID and 30 controls. The research is in press in cardiovascular diabetology. Some of our most noteworthy findings include a downregulation of the molecule kallikrein, an upregulation of platelet factor 4, once again von Willebrand factor, and marginal upregulation of, again, alpha-2 antiplasmin. If you return to the clotting pathway diagram, you can see where these molecules play a significant role, and all points to decreased fibrinolysis and eventually fibrinolytic failure. We also found a significant presence of antibodies this time entrapped inside the microclots when we searched for it. I just wish to return to some of the latest research from other groups. Here is a document from the US government where microclot presence, viral persistence, autoantibodies were recognized as central pathologies to look into in finding answers in long COVID. And this is very important. I'm also happy to report our work was featured in Science in June, as well as recently featured in Nature. And to conclude, just the way forward, together with our team of international collaborators, we will report back soon on more laboratory results for long COVID. Researchers at Sheffield Hallam, as well as University of Manchester, have confirmed microclots in long COVID patients and could correlate the degree of microclot presence to disease severity. Dr. Benita Khan and Dr. Ray Duncan, which talked before me, are involved here. We desperately need clinical trials on treatment that combine prevention of microclot formation and platelet hyperactivation. Treating only the one and not the other will perpetuate vascular damage. And just my final comment to report from the WHO panel meeting earlier this week, where I was a panel member. We are currently formulating the case definition for long COVID in children. Thank you for allowing me to share our research with you.